Yeah. Using this Eminem freestyle, 8 Mile, instrumental. Smartest, double helix. Born in 1916, I'm Crick. My double helix pride says I'm no hick. 21 years, receiving my back strike in physics. During the war, I made this mind. Check it out. It's sick. Since the war, I was all about molecular biology, totally unrelated to my study of water viscosity. I started looking at cytoplasm and all that, examining protein structure, which I would later benefit. Moving on to X-ray crystallography, taught it to myself. You won't find it in my bibliography. All this biophysics cuts like a knife on the way to the secret of life. It didn't take a PhD for me, Francis Crick, to see that the hypothesized alpha helixes will not function properly. On to the course. A to the T and C to the G. Take a gander at all of our degrees. It's James D. Watson and Sir Francis Crick. We got the discovery of the double helix. Yo.
Straight out of Compton. Born in 1928, his work with Crick has made him great. Call me James D. Watson. At the tender age of 15, I got some education at Chicago's University, age 19, BS in zoology, that's Bachelor of Science to you. Not the other derogatory term you may be used to. Early 1948, I joined the phage group. I soon found myself in a scientific troop on the way to finding the secret of the gene. Back in those days, we thought the genes were proteins. Protein genes, whatever could they mean? I was just looking for some knowledge, and green DNA was thought to be a stupid tetranucleotide. From the phage group, I knew better. I soon cast his guess aside. Avery and his colleagues conducted an experiment. This overlooked DNA and turned out to be important. Word, word. What? Split in.
Splice.
Splicing.
Whatever you want.
Splice. Splice. Splitting.
Clean and Marcus.
Dr. Crick and Watson. October of 1951, legends get together.
Check out my belt. It's made of faux leather.
Dr. Crick, that was quite a pathetic rhyme.
Quiet, Dr. Watson. I'm just trying to fill the time.
Now we know DNA holds the genetic instruction. We snuck a peek at Rosalind Franklin's X-ray diffraction. Our first model of the phosphate backbones are on the inside. No, the phosphates are almost certainly on the outside, Miss Franklin replied.
After our first failure, we were then forbidden, like Adam and Eve in the Garden of Eden. 1953, we got another chance to try again. More of Franklin's finding we did secretly obtain.
Playing with molecular models made of cardboard. Wait, they fit. Something struck a chord.
Watson, we did it. The chains of nucleotides.
There's two of them in a double helix. They entwine.
On the inside, you can feel the nucleotide base.
With the phosphate backbone, they form a helix staircase.
Number of adenine and thymine are in proportion. Same with cytosine and guanine. There is no distortion.
Adenine and guanine are purine, cytosine, and thymines, pyrimidines. Well, what do you know? They have the same ratio. Well, now break it down. AKFC's Patty O.
AT base to get two hydrogen bonds. GC got streak. Dang it strong. Two ring pyrimine binds with one ring pyridine. This helps keep the diameter to approximately 2 nanometer.
These two nucleotide strands are complementary. You'd think that this stuff would be elementary. ATGC is base paired with TAGC. Well, how about ATGCC? Let me try that again. Well, how about ATGC TAGC? That would be TACG ATCG.
All of these strands are running 5 to 3. This is a typical of the time. But they're going in opposite direction. This is anti-parallelism in our perfection.
This anti-parallel configuration holds the implication for future generations. It's the key to replication of DNA to avoid mutation.
Coming at you, live, the master of disaster, run and hide. Every single day, we reppin' double helix. Genetic history, son, we bout to reveal it. Every day, we reppin' double helix. Genetic history, son, we bout to reveal it. Let me spit you some knowledge so that you can understand it. It turns out DNA is really double-stranded. This was published in '53 by Watson and Crick, which was a big discovery for all of genetics. Then, in 1959, a scientist known as Jerome probed an abnormal human chromosome that can ultimately lead to Down syndrome. For work in DNA, it was no surprise that Watson and Crick received the Nobel Prize. And Rosalind Franklin, even though she died, received very little credit because she wasn't a guy. In '82, a genetically engineered drug known as insulin was marketed by Lilly called Humulin. We reppin' double helix, yo! We reppin' double helix.